The Molecular Biotechnology Laboratory at Addis Ababa Science and Technology University is a well-equipped facility supporting research in genetic engineering, molecular diagnostics, and bioinformatics. It provides advanced tools for DNA, RNA analysis, gene editing, and protein studies, benefiting students, researchers, and industry professionals. This lab enhances AASTU's role in biotechnology innovation and applied research.
My name is Dr. Kizacho Haile. I'm one of the biotechnology staff and the head of this laboratory. I am a GEO, sometimes we call it Molecular Biotechnology Laboratory. So we provide services, especially experimental services in molecular biology for undergraduate and postgraduate students. And also we provide services for other communities as well from other universities across Ethiopia. And even in our previous time, we provided support for Ministry of Health as one of the COVID testing laboratory in Addis Ababa. So both researchers and students can utilize these facilities. We have very interesting facilities, like this area is DNA extraction area. So we have eating walls, we have centrifuge, autoclaves. And there is a sonicator. This is for disrupting cells. So it works by generating a sound, high frequency sound. And this is also we call it tissue-lyzer for breaking hard tissues. So we utilize it for especially plant and animal tissue-lyzing or for tissue restoration. This is a centrifuge. And we have a plate, which we call a plate reader. So if anyone is interested in reading ELISA plates, so they can put the plate here and visualize the color change. This is very important for immunological studies. If someone is interested in immunology, so for immunological studies, especially antigen antibody reactions, so they can utilize this. This is a spectrophotometer. So the UV spectrophotometer. It's also for absorption studies. If anyone interested in cell turbidity or for DNA quality checking studies, so we utilize this machine. We have machines here we call PCR machines. So these PCR machines are very important. We utilize it for amplifying DNA, for copying DNA, or multiplying DNA. So we have some 4 machines. We have also some other in other laboratories upstairs. These machines are very important for cloning or multiplying DNA, or amplifying DNA.
Okay.
Then after amplifying DNA, we will take the DNA to visualize in gel. We have the gel. This is we call a gel electrophoresis unit. Of course, we have a small type and medium type. This is a small type. So this is one of the voltage supply system for the gel. This is a gel unit. So we prepare a gel and put the DNA and finally allow the DNA to run. Then based on the size of the DNA, it separates the DNA. So this is one of the DNA visualizing equipment. Then after DNA visualization, after gel electrophoresis, we will take it to gel documentation. I will come to that about the gel doc. We have also another electrophoresis unit. This is we call vertical electrophoresis unit. So this is very important for protein electrophoresis, the small type. We can extract protein, we can run again the vertical. These are the glass plates, accessories of this one. And we have also another types of the vertical. So this is for vertical electrophoresis. Then after vertical electrophoresis or horizontal, we will take the DNA or protein to a machine called gel doc. This is a gel doc, gel documentation machine. So we use this for DNA documentation. After gel electrophoresis, we'll put our sample here. sample here, and finally we will visualize it. So this is, we call it gel documentation system. So we have a mini computer, we have imaging systems, and we use this for visualizing of gel. Again, this is the gel documentation system. We can use this for both protein and DNA. Again, we have a camera system and we have a UV system. So by utilizing this UV and camera, we will use for documentation. We have another small, this is also a gel documentation system. So first, while manipulating this, we will wear a mask. We have a face mask for protection of UV. So we will put gel here and we'll on the UV system. With this we call the UV trans illuminator. This is the earlier type of this machine. This is very old and the earlier type of this machine. And this is again utilized for visualizing the gel. And we have another facilities. So we normally work with chemicals, fuming chemicals. So this is one of the safety protection for anyone who is working with fuming chemicals. In molecular biology, we manipulate chemicals like chloroform, formaldehyde, and other chemicals. When they are fuming chemicals, so this is very important as protection equipment for anyone who is working with chemicals like this. And we have also freezers for keeping our samples and DNA. The freezers as well as minus 20 and 4-degree freezers. Okay. So this is one of the very important equipment that we have in our laboratory. We call it the quantitative PCR. So this is the machines that we have been using for COVID testing in our previous public one of the COVID service that we provided in our previous years. So this machine is very important. It serves many purposes. One is for routine RNA quantification, we can utilize it. Again, for DNA quantification, if we have any DNA sequencing system is there, we can utilize it for DNA quantification. Again, we can utilize it in forensic science for identifying DNAs, and again for diagnostic purposes that we have provided in our previous years. That's one of the common service analysis. So this is very important machine. It quantifies DNA. Again, it detects any possible virus or bacteria that is present in human samples. So for detection as well as for quantification, we can utilize it. This is one of the very important and very expensive machine. It's again very important if someone is interested in extracting DNA from pathogenic viruses as well as from pathogenic bacteria. We have a safety system we call the biosafety cabinet. This is a biosafety cabinet, and this cabinet is very important because it sucks air, any contaminated air, and finally release clean air to us. So this protects anyone who is working with pathogenic bacteria or viruses. So this is a safety equipment. So we utilize it for again DNA extraction. If someone is interested in extracting DNA from pathogenic bacteria or virus, we can, or he can or she can utilize this facility. So we have again heating plates. We have again microplate readers. We have extraction equipment like tissue lasers, and we have centrifuges as well. So this facility is also a very important facility to use and work in molecular biology and biotechnology. So anyone is interested in utilizing these facilities or our facilities, please contact us. So we are here to serve you in ASTU. So thank you very much.